Hello, and thank you for joining us. I am Jasmine Navarez, the Digital Content Coordinator at Simulations Plus. I'll be hosting today's webinar behind the scenes, making sure we don't have any technical difficulties. In this webinar, Dr. Hovik Gukasyan, Associate Professor of Pharmacology and Pharmaceutical Sciences from USC, and Dr. Maxime le Merdy, Director, Research and Collaborations from Simulations Plus, will discuss how modeling and simulation platforms and the GastroPlus Ocular physiologically based pharmacokinetic model in particular are supporting researchers in overcoming ophthalmic drug product challenges. Dan O'Connor, our Senior Director of Business Development, will serve as our moderator today. A few housekeeping items before we begin. We take your privacy rights seriously. By registering for and attending this event or participating in the Q&A session, you are allowing us to contact you for follow-up. You may ask questions via the questions panel on your dashboard at any time. If you need assistance, please use the hand raise icon. We'll address your questions in the Q&A after the presentation if time permits. This webinar is being recorded and the replay will be posted on our Simulations Plus YouTube channel. And I'll go ahead and pass it over to Dan to introduce our speakers.
Good morning, and thank you very much, Jasmine. Good morning, good afternoon, good evening. My name is Dan O'Connor, and as Jasmine mentioned, I will be moderating today's session with Dr. Max le Merdy, who's calling in today from Geneva, Switzerland, the home of the second best chocolate in the world, Hershey, PA, of course, being the best, right, Max? And then he will be handling the downstream application for ocular modeling as it looks to take the data from a preclinical into the clinical setting. He will be the second participant in our webinar this morning. He will be preceded by Dr. Hovik, whose last name, if I pronounced it, I would destroy, but I'll give it a shot. Gukasyan is Associate Professor of Pharmacology at USC. He just told me moments ago that he rides the train every day from Orange County up to LA and back. So he's welcoming all of your questions, which if he can't answer on this call, he'll be able to do it on his way home from LA this afternoon. So without further ado, let me turn it over to Dr. Hovik. Why don't we get started and help our web attendees learn more about how ocular modeling can help advance their research.
Thank you so much, Dan, for that great intro. Really appreciate it. Hi, everyone. Very happy to be here this morning. This is a special treat. I appreciate Simulations Plus for organizing this webinar. I'll be talking about in silico methods for ophthalmic drug discovery and a little bit of product development today. And I think hopefully I'll set up my friend and colleague Max to talk about his stuff nicely with this. So I'll be focused more on early stage concepts that I work on with my students at USC. And if you want to connect with me, I got a little QR code there for a LinkedIn connection. Don't be shy. I'd love to keep in touch with everyone, whoever is interested in what I do. Some of this work has been presented at several meetings earlier this year. Most recent one was at an ophthalmology meeting called ARVO. The sponsor of the project is Boehringer Ingelheim. I do want to acknowledge them as well. We do have a research grant, research/educational grant from Boehringer Ingelheim to explore this concept and possibly find some useful molecules. And I'll talk about that very process today. And of course, Simulations Plus with their University Plus educational research consortium program is really the last pillar in making this possible and allowing me to give this talk today, also giving us access to their platforms like ADMET Predictor and GastroPlus and OCAT, right? So you probably can't read the names of the authors in that little snippet there, but I'll show them I'll show you guys who they are before I start talking about my story, right? These are the students that I worked with to do this work. They did all the work, actually, the heavy lifting. They're passionate about drug discovery and drug development, and I am really happy to have them work with me. And they've learned how to use these in silico tools very well in the past year and a half. That's Andrew up there. That's Abby, Ethan, Bruno, Lilith, and Tiffany. And this is a picture we took at Arvo where they presented all of this work. Actually, you're gonna see about 5 months ago. All right. Our group is called the USC Men's School of Pharmacy Drug Repositioning Group. And just in case you've never heard of this concept called drug repositioning, or sometimes people call it drug repurposing, is we try to identify new indications for existing drugs or molecules that are not even drugs. They may be discontinued or shelved assets, or molecules that are currently under development for other conditions. If possible, we can access them. So this is more of a disease indications discovery effort rather than a molecular design effort. And there's a lot of substrate like this out there, and I'm gonna talk about only one source today, which comes actually from the sponsor, Boehringer Ingelheim, a source for substrate like this. And I really am passionate about this approach because I feel it maximizes the use on all the prior and current assets that organizations, sponsors originally made these compounds for, with probably different objectives in mind. So we're gonna just take the molecules at face value and try to focus them in ophthalmology. That's essentially what we do in my group. So before I get into the simulations and in silico tools, right? Just as an outline, want to point out that the platform where the compounds come from is called OpenMe, and the website is also called openme.com. It is curated and managed by Boehringer Ingelheim, so they own these compounds. And at the time the students started working on this particular cycle with me, and the compounds always change, right? They update them, they take some off, they add more things to the website. So we accessed this in 2023, right? There at the time we were able to look at 58 different compounds on this website. That's a lot for 5 people, or 5 or 6 people. So, and we looked at all of them, right? First, what we did is apply. This is the outline of the presentation. Is we applied this concept of drugability characteristics for topical ophthalmic drugs, called the rule of ophthalmics, ROX. To see if they are all suitable for topical ophthalmic delivery. Again, agnostic of an indication at this point. That eliminated only 12 compounds, which is a good percentage, right? I think the more you eliminate, the better, actually. And then we applied our second concept, and I'll talk about that. Pardon me, before I move on, I'll talk about the rule of ophthalmics and this drugability criteria in the context of ADMET predictor. That's the platform we use to study these properties. Then we just bring in a simple literature research component at that third phase, where we look at essentially just the pharmacology, or the targets, protein targets in the body for the remaining compounds in that subset, and try to match those targets with as many reports of a role in a specific disease that we can. And the more hits we get there, again, the better and if the reports are actually coming from different groups, different universities, that's even better, if versus only one place is constantly publishing about them. So we essentially try to validate these links of the targets of that the molecules either inhibit or activate to a disease in a ophthalmic setting, right? And then at the end there, we actually bring in OCAT at that point once we find good targets matched with good compounds, and we develop a formulation for those compounds for testing. And this is where I'll end my talk, right? And we have identified 5 promising leads from 58 to date about this concept, and we're working on all of them at the moment. This is work in progress, but I'll give you one of the case studies, and you'll see how we have utilized OCAT to prepare one of those 5 compounds for a PK/PD experiment, PK or PD experiment. So these are the rules in that second layer of the filter that we applied to all the molecules that we downloaded from openme.com, all those Boehringer Ingelheim compounds, right? There are 4 rules, and all of them, except for rule number 3, are actually captured in admet predictor. So simple, if you've used admet predictor before, you can run these calculations very efficiently, and you'll get your log D values at pH 7.4, your topological polar surface area, the intrinsic solubility. So they're all there. The only value that we had to input manually, which also admet predictor allows, is this partition, pardon me, this energy change, free energy change of partitioning between an aqueous environment and the plasma membrane. This is how we visualize this delta G molecule. And any compound that complies to these boundaries or falls within these limits has been actually successfully developed into a topical ophthalmic drug product subsequently. That's where the data comes from. So this is gonna bias the selection from Boehringer Ingelheim towards topical ophthalmic delivery. Here is the data we generated. This comes right out of admet predictor 10. I believe we used admet predictor 10 back then. There's a newer version available right now. But this is a terrific tool for drug discovery. I want to mention, right, again, we have, if you add up these numbers on the column heads, we have 58 compounds, as I promised before, so they're all captured here. It's not a big data set, but it's quite a lot of different molecules with different targets to sift through, a lot of pharmacologies. And here we're just showing the distribution of all of them as a function of these 4 druggability attributes for topical ophthalmic delivery, right? You have solubility here at pH 7.4, the distribution coefficient at pH 7.4. You have topological polar surface areas, and also this free energy of partitioning between aqueous environments and plasma membranes. And we eliminated, I recall I said 12 compounds through this process from 58. So it narrowed down the list quite a bit. And then we matched the remaining 12 compounds with targets in the eye that are linked in the disease to a specific ocular disease. The way these druggability criteria were crafted, they all relate in some way to optimized corneal permeability, essentially, these properties, right? They make either the drug more available for absorption into the cornea, or they are intrinsically linked to the property of permeation, passive diffusion through the plasma membrane of a cell monolayer, right? And this shows like a 3D distribution plot of the best compounds from that data set. And actually the 5 molecules that we selected to work on, they are also captured in this data set. And you really want to be sort of in the front right-hand side of this 3-dimensional space, in our opinion, where the dots are. That's the best space to be, because it shows you on that x-axis the log of the corneal permeability. This is also an ADMET predictor calculation. You want to make that as high as possible, right? Preferably the y-axis, the vertical axis, which is the solubility of the compounds at neutral pH. You want that also to be very high, because that's desirable for topical ophthalmic dosage forms. And then the distribution coefficient, right? You want that to be somewhere in the middle of that range, ideally, not too hydrophobic, not too hydrophilic, somewhere in the spot in the middle. So compounds around here, if you can see in 3D, are the best chemical space, in our opinion. And just for fun, we use the ADMET predictor to color code them to compare these rules of ophthalmic drug-like criteria to Lipinski's rule of 5 that everybody knows about. And interestingly, there's one compound that really breaks Lipinski's rule of 5, just one, this red one. But it falls actually in this space that suggests if it might be a horrible oral drug candidate, it might be a really good topical ophthalmic drug candidate. It's interesting to see that. The yellow one breaks 2 rules, the green ones break 1 rule, and the blue ones do not break any of the rules. But they all pass the ophthalmic rules. Right? I'm going to talk about of course, the winning compounds there, but I do want to point out the failures, because this is a really good learning for us from ADMET predictor on how the software helped us apply these concepts in early lead selection in a drug discovery process. And as you can see, a lot of the compounds, they look quite large by size. If you are keen on looking at the structure, I mean, they are just monstrous looking molecules, a large molecular weight, right? And a lot of them have a extreme in solubility. For example, they're either extremely water soluble, or they are not water soluble at all. They have very high distribution coefficients at pH 7.4, and they violate even more Lipinski's rules. So these are the failures. So some, for example, there are many ProTac technology compounds on openme.com, and we took all the compounds. We didn't pre-screen them or anything. So obviously, if you know about ProTac, yes, that falls in here, right? These are typically very large compounds, and there are a couple of them. This compound 3, also compound 8 in this list. I think there was another one, maybe compound 7 as well. So these were the ones that we sifted out. So now, towards the last few minutes, I want to shift gear on how we take the winning 5 compounds from ADMET predictor, and then use OCAT with GastroPlus, and continuously interacting with the open source repository from Boehringer Ingelheim for information to build models and use those models to design prototypical formulations for non-clinical testing and proof of concept work. And here I like to rely on some prior publications that I like to read. So I want to share it with you guys. This Vezina paper about comparative ocular anatomy in lab animals is one of my favorite papers. So that's panel A there is the primate, the B is a dog, C is a rabbit, D is a pig, E is a rat, and F is a mouse eye. And you can see how different these species are. And probably if you look, you'll find ocular drug discovery or ocular pharmacokinetic studies using any or all of these animals. So you have to be aware of their ocular physiology, geometry, and anatomy if you want to effectively use model-informed concepts to design formulations and delivery systems for testing of new chemicals. And the other work that's absolutely seminal in this context is these publications from Max, actually, my friend Max le Merdy, who'll speak after me. But he really set up a series. I'm citing one of these papers here, but he has several publications that exemplify very nicely on how you can utilize ocular compartmental absorption and transit module within GastroPlus to simulate ocular pharmacokinetics. But I think more importantly for me, because I'm a pharmaceutical scientist, not really a PK person, the effect of dosage forms and formulation design on ocular pharmacokinetics, because that plays a huge role. And just remember that you have to link the GastroPlus section, the ACAT section, right, to the OCAT section. They must be connected, right? You can't look at the eyeball in its own universe without considering the rest of the systemic circulation and the central compartment. So they're connected, and I'll mention this in the example that I'm going to talk about next. So I don't think I have time to go over all 5 compounds, but I'm happy to share the other 4 that I'm not going to present today because of time constraints. I'm going to talk about one of the 5 winners that basically catapulted from ADMET predictor to OCAT GastroPlus, right? This was a compound called BI113823. I think Tiffany was the pharmacy student that worked on this particular case study. It is a bradykinin 1 receptor blocker or antagonist. So that's what the compound does, and it happens to actually be reported to play an important role in modulating the permeability of retinal vasculature. So many researchers think that, and this convinced us too by reading their papers, that blocking bradykinin 1 receptors may play a role in important diseases like diabetic retinopathy, complications of retinal physiology and retinal disease as a result of type 2 diabetes. A lot of people suffer from this. So we move then the compound from ADMET predictor with its druggability criteria summary to OCAT GastroPlus, and then we bring in support parameters, right? When you build a GastroPlus model, in an OCAT model, you need several critical descriptors of the compound properties, the compound's pharmacokinetic properties, its biopharmaceutical properties, and species and PK data in order to set up a model in GastroPlus that will be accurate or predictive. And this particular compound actually has a lot of mouse and rat data available from openme.com. Well, let me show you what it looks like, right? So you start building your model. You refer to openme.com and pull out the certificate or for sort of like the birth certificate document for 113823. They have these things for every compound that's there. It gives you this data. So they have mouse PK parameters and rat PK parameters, and they tell you what the dose and the route of administration was, right, for each of these studies. Not every one of the 58 compounds has this much non-clinical PK data about it. So they're not all. You're not going to be this lucky with every compound there. This is, I think, it's a lot of data for a shelved asset, I want to say, with no purpose right now. So we take this data. And we also take the compound properties, right? PhysChem, biopharmaceutical, formulation, in vitro, ADME properties. These are also all published in openme.com. Very important input parameters for building a GastroPlus OCAT model. Like they even have permeability values, right? From a couple of in vitro studies, KCO2, MDCK. They give you the human, rat, and mouse microsomal or hepatocyte extraction ratios there, plasma protein binding. I mean, these inhibition constants for CYP enzymes or the hERG potassium channel, they're not that important for ocular indications, but it's good to see that the compound is decent from a systemic exposure safety point of view, right? I just maybe leave it at that for now. Right. So then the very first thing we do is we try to set up a model for the openme.com data that's provided for us. So we did two GastroPlus models in this case, one on the observed mouse PK, right, and another one on the observed rat PK. And you can see without a lot of optimization, essentially just taking the numbers from Boehringer Ingelheim and their compound and plugging them into GastroPlus using a non-clinical species for PK prediction. We got a pretty good, I think, agreement between the observed and simulated values. And if you spend some more cycles to optimize these values, the model will become highly predictive, right? But that was not our goal, right? We are not gonna use mouse cancer models or something else, or rat models with systemic dosing to push this molecule forward. We are focused on ophthalmology, so this was not optimized too much further. At this point, we actually shifted and we created two different comparisons with mouse ACAT data and rabbit OCAT data, and then rat ACAT data to rabbit OCAT data. So like we used the mouse data and we came up with a suitable dose and a theoretical formulation for this compound. There's a lot of background work here you don't see, but the simulation here shows a hypothetical 30-day, 1-month study where you reach a steady state after dosing this compound. And that steady state, it's way above IC50 value for the bradykinin 1 receptor, which is about 7 nanomolar. It's a very potent compound, but you do reach and what we call IC99 is 10 times higher than the IC50 value. We just arbitrarily decided to go 10-fold higher in terms of exposure. And the blue lines are in retinas, and the orange lines are in the choroid, because this may be useful for diabetic retinopathies, right? When you do the rat to rabbit transfer, again, this from systemic dosing to topical ophthalmic dosing, just the time to reach to the steady state exposure in after a month of dosing is a little bit different, but it still gets there, right? So we think that now this is good to confirm in a prototypical formulation in a rabbit ocular PK study to validate it. And there are several rat and mouse, actually, diabetic retinopathy disease models that we can use for at least a month to see if the compound actually will give you efficacy in those models as well. And before I conclude, right, I think one thing, well, maybe I can go to the next slide here. These are very good comparisons of eyeball sizes and shapes for various species that we talked about. So we had the 2 rodent eyes here and the 2 rabbit eyes. But pragmatically, right, I think what GastroPlus OCAT really helps you with is the ability to look at exposure in tissues like the retina or the choroid in these really small species, which are very hard to do bioanalysis and PK studies actually on rodent eyeballs to get drug exposure levels. But you can actually get some estimates to help you in your formulation design or your experimental design of using the software. So it's going to be very highly model integrated and model informed in a sense. So this is my last slide, but like I mentioned, this is MIDD, in my opinion, or model-based drug discovery and development. We are gonna try to put this formulation that we came up with now, which enables those two PK profiles that you saw, at least in silico, in a retina or choroidal tissue, and test that. In a rabbit pharmacokinetic study, and we're looking for a collaborator that has the diabetic retinopathy rodent models to test this formulation for us in a efficacy study. And once we get the data back, we will go back and actually fine tune our OCAT model to make it more predictive, right? This is completely predictive right now. And I used AI actually to generate that picture of a eyedropper bottle with the Boehringer Ingelheim number on it. So that's not a real eyedropper. I asked artificial intelligence to make it for me. I thought it was cool, very future looking. So I'll stop now and hand it over to Max.
Thank you, Hovik. So let me share my screen.
Do we have a poll question, Max? I don't know if Jasmine has a poll question.
Yeah, let me go ahead and launch it now. Oh, that's not it. Sorry. Give me 1 second. Okay, it's basically just asking if you're interested in having somebody follow up with you about leveraging the OCAT model in GastroPlus. And I'll give everybody a couple seconds to answer that. There.
Okay, so let's get started. So Hovik actually finishes his talk with mentioning that needs data to further verify his model. So what I'm going to present today is actually when we have some data and how can we leverage those preclinical data to predict what's happening in human. So very briefly, because Hovik has introduced it, but this is the structure of the OCAT model. And you can see how we are covering all of the ocular tissues, and Hovik has talked about the retina. And you can actually predict the retina concentration following topical administration. And the model is covering different mechanisms such as diffusion, convection, the aqueous humor flow, as well as a transfer between the ocular tissues in green and the systemic circulation. So we have all of that. A very brief history about the model. This model was initially created in 2010, and the first version was a very simple model that integrated 6 to 7 tissues. And then between 2010 and 2020, we have made very significant improvement to the OCAT model structure to end up with the one I've just presented. And also, as Hovik elucidated already, validate how the model could predict formulation modifications and its impact on rabbit PK and also on human PK. So we have made extensive work with a solution, with suspension, with ointment. And I would like to invite you, there are a couple of publications here. You can type my name in PubMed, you will find them. We have tried to publish all of the results over there. In 2020, we had another collaboration because something I would mention is we actually started to work in collaboration with the FDA on development of this model in 2014. In 2020, so 4 years ago now, Simulation Plus has been awarded a new grant to really focus on that interspecies extrapolation and how can I predict the PK and or the PD, pharmacodynamic, in human after my model has been validated in rabbit. And that's what I'm going to talk about today specifically. So once again, during this collaboration with the FDA, we have identified a methodology on what would be the best way possible to do that rabbit to human extrapolation. And we have defined that starting with New Zealand rabbit or albinos rabbits, and then moving to Dutch-belted to account for the effect of melanin would be the best way to calibrate the model in preclinical species. And then switching the physiology from rabbit to human would help to predict the PK in human. So that's the overall strategy we've been developing. What I'm going to talk about in the next step are 3 case studies. One for solution, where we extrapolated the PK. One for suspension, when we also extrapolated the PK. And finally, the last one also for suspension, but this time we looked at the pharmacodynamic. So the first case study actually that I'd like to talk about, and those have been published last year, I think now, is for solution. And the first question we asked ourselves, which was now 4 years ago, was with the model that I have, and once it's been validated in a rabbit, can I predict the human exposure? And we started with the simplest solution, formulation, aka the solutions, so we didn't have to deal with the formulation aspect. And for this work, we looked into 3 drugs. One of them was gatifloxacin. We also had mofloxacin and levofloxacin, but I'm just focusing on the gatifloxacin in this slide. And on the left here, you can see how the model in the top has been developed using cornea and aqueous humor data in rabbit, in New Zealand rabbit, and then also doing some validation in Dutch belted. Once we had the model developed in rabbit, we used it to predict different scenarios also occurring in rabbit, all of them in New Zealand in this case, whether the drug was administered once or multiple times. And you can see that overall, the model is doing a reasonable job at predicting this data. One thing that jumps directly to our eyes here is there is a very significant interstudy variability. And that's something that is known for rabbit PK study, ocular PK studies. So also something we had to integrate in our mindset when we were to develop and validate these models, like accept that not all predictions would be perfect. Because to give an example, these two studies are coming from the same labs, just 1 year apart, using the same drugs and the same rabbits, and yet the results are quite different. So this is what we had. Now the model was validated in rabbits. We then moved it to human, and we had data that has been collected in human in 4 different states, either patient with cataract disease or patient that had to go through keratoplasty, vitrectomy, as well as healthy subject. And you can see that overall, all of those are just pure predictions, which means that we took the model developed and validated in rabbits, we changed the physiological parameters of the eye from rabbit to human, and then we predicted how the drugs would behave in the human eye in different tissues. So you have in red aqueous humor, in green the cornea, in orange here or light brown the vitreous humor, and then in purple and orange the conjunctiva. And you can see that once again, overall, the model is doing a reasonable job at predicting the observed data. One thing you can see again is for some studies, the error bars are very significant. So we are expecting a very large inter-subject variability. So one drug is not enough to qualify whether the model can perform this task or not. So we actually did the work for 3 drugs, and you have here the summary of the data that we had. And once again, overall, the model, if we look at those, I think 25 plus studies, the model was able to do a really good prediction of the observed data without modifying the model between the rabbit and the human phase predictions. So we were happy with the solution. We had done other case studies I'm not showing here. And in the end, I think we've worked on 7 to 8 solution case studies, and they were all very positive for the extrapolation. So then the next question was, can I go a step further and increase the complexity? And can we actually predict human ocular exposure for suspension? And we also published an article earlier this year focusing on this suspension. And we actually work with a drug named Bezivance. And this drug has a specific characteristics in a way that it is formulated with an excipient called Duracite. And that excipient, what it is going to do compared to what we see in a normal suspension is create a gel-like polymer. I mean, the polymers will create a gel-like system that will layer the surface of the eye where the drug can be entrapped, and it affects the overall residence time on the surface of the eye and therefore the exposure. So in order to model this, what we've done is account for, we actually model the suspension as a mix of a solution for the dissolved fraction and a topical control release formulation that would account for this effect of the excipient on the residence time on the surface of the eye. And this is what you see here in this plot. You have in the dashed line the simulation if we were to simulate the drug as a suspension, and the solid lines are the simulations once we account for the effect of Duracite. And you can see at the bottom the final fitted release profile for that. But once again, one study, so not enough to validate. So what we wanted to see is, can I validate the model? And for that, we had 3 studies in rabbits, one in New Zealand and 2 in Dutch belted. And once again, without modifying anything, you can see that overall, the model is doing a reasonable job at predicting the data. So we were fairly happy with that. And now what we've done is we went from rabbit to human, and you can see that once again, the prediction in human for the tears, the conjunctiva, and the aqueous humor are really good. And for that, we kept all of the drug-specific and the formulation-specific parameter the same, meaning that the release profile that we had fitted in rabbit was also used in human for the predictions. So we are happy with the suspension prediction as well. So we did also additional case study. So we are really confident in a way that we could actually predict the PK in human for multiple formulation once the model has been heavily validated in rabbits. So once we wanted to increase the complexity again, and we start to look at pharmacodynamic. I'm taking the case for suspension, and I'm going to talk about brinzolamide. So here on this study, you can see the PK data in cornea, conjunctiva, aqueous humor, and ICB, with data coming from 4 studies. And because we had a significant variability, I've tried a different approach in this case study, which was to pool all of those studies together. And instead of doing single simulations like I've done in the previous case study, is actually to go to a population approach. So what I've done is I've calculated the total numbers of rabbits that have been used in those 4 studies, for example, and then created a virtual population using the software that was able to represent that the observed data. And this is what you can see here, and I just focus mostly on aqueous humor, but you can see how the model is able to describe really the shape of the profile, that's one factor, but also reproduce the observed variability, not just within one study, but also across the different studies. So we had the PK model validated in rabbits and set. So that was good. So now looking at the PD, we know brinzolamide will limit the production of aqueous humor in the epithelium of the salivary body. So I wanted to have a PD model that can describe that part of the mechanism of action. So it's a bit tiny, but you can see here on the top how the aqueous humor concentration of brinzolamide will have an impact on the production of the IOP. And we had developed a special IOP model that is soon to be published. But long story short, you can see in this slide how in green the PK in aqueous humor and what would be the resulting prediction for IOP reduction in rabbits for a 1% dose. And you have the model parameters here. Interestingly, the variability we saw on the PK side was also observed on the PD side. And I had collected 9 studies in the literature looking at the PD effect of brinzolamide 1% using all the same drug product, which was azopt. And you can see there were very significant variability. So instead of looking at just one average prediction that could describe more or less each individual studies, I've pulled everything together and did this population approach. And you can see how the model is able to describe fairly well the reduction in IOP and then the slow return to baseline, and also capture the variability across all of those different studies. So here I have this population PBPK PD model validated in rabbit. So I kept this one, I use it, and I wanted to look into the human data. And once again, here we had collected lots of data in human coming from different studies. And you can see we had different doses, we had different administration schedules, either twice a day or 3 times a day. And we had also different study length going from 2 weeks to 18 months. And once again, instead of looking at these data one by one, I've just pulled everything together. And you can see here the pharmacodynamic prediction for IOP reduction of ASOPT in human using the PK, PBPK, and PD model validated in rabbit, and how the model is able to predict really well the shape of the profiles. Like you can see how we have this initial elimination and then this plateau. It's a bit harder to see, but we have also the steady state prediction that is really good. And we're able to predict the data after 1 week, 2 weeks, a month, 2 months, 3 months, and 6 months really well. And we did that for the BID administration, and we had similar results for the TID administration. And you can see once again, the shape is slightly different because we have more administration. The steady state level is also slightly different, and we're able to capture that. And you can see that once again, all the way to 3 months, we are able to have really good predictions. So with that, I just want to thanks all of my colleagues at Simulation Plus, so Viera, Jessica, Jim, and all of the research and development team that helped me in this project. I also want to thank the FDA colleagues, especially Dr. Minliang Tan, that is the project officer on the FDA side that has really helped a lot the project. And if you have any question, happy to connect with, if you want to connect with us, you can reach out to me, you can reach out to us, and happy to help. Thank you very much.
Thank you, Max. This is Dan again. And we've had a couple of questions that have come in the background, and they've been answered. I don't know if everyone can see the answered questions, but we would welcome additional questions that can be typed in real time. And I'll start with the first, if I may, excuse me. The question is wondering which parameters were incorporated the variability in order to perform population simulations with rabbits. If you assume the same CVs for all of them, or is there any literature source available with this information? That's directed to you, Max.
Yeah, so oops, sorry. So that's actually a great question. So far, and that was one of the optimization optimization aspect that we did as part of this project. I don't have it in the slides, but we had variability on the IOP influx and IOP efflux parameters. We also had all of the variability coming from the PK side that is already by itself influencing the PD. So that's one factor. And then we had assumed a similar CV% for all of the PD parameters. That's why this work is not published yet, because this is something we're revising, whether we can identify in the literature, for example, the influx IOP parameter. Can we find the literature a value and see whether this value would work and be able to reproduce the data you see on the slides? So far we had a non-mechanistic approach for the variability, and we assumed 15% variability for all of the parameters, and that works quite well. This 15% is not coming from anywhere. It was actually the observed variability in IOP across all of those different studies, and at pretty much any time point for all of the studies, the variability was between 15 and 20%. So that's why we started with that, but we'll try to refine the model in that way.
All right, thank you very much. We just had another question. Well, let me ask a preceding question. And it's about validation. Have any of the models been validated for a preclinical species?
Yes, so actually, as you can see on the slide, for rabbit, we've done extensive validation. We have also worked with the monkey, rhesus monkey, and we had a poster on that I would be happy to share if you want. So the monkey is validated, and we recently worked on the dog, especially for more complex formulation like insert and implants.
Okay, great. And I see that people are attending, a few of them have left. So let me just run a commercial here real quickly. Hovik is a great testimony to the University Plus platform that we offer to any validated or accredited academic institution where we provide all of our software at no cost. So if anyone on this call is in an academic institution and seeking the same platforms, or if you know someone in an academic institution that might be advantaged by having access to these platforms, just let us know. And Hovik, maybe I could ask you the question, how has the adding modeling and simulations to the curricula for your students change their perception of drug discovery and how they approach it?
Thank you, Dan. It has had a tremendous impact on the students' excitement level about drug discovery and development. I think one aspect is that myself and several of my colleagues at USC as well, we use GastroPlus through University Plus to teach the students concepts on uh, uh, PK or concepts on drug design and optimization, and they see it happen on the computer screen, and it just makes it all very much real, especially for young students that have never been exposed to these besides like basic sciences. So it's a terrific tool to teach besides do directed research, I think.
That's great. All right. Well, that was the end of my commercial, but thank you very much for sustaining it, Hovik. We do have another question about specifically ADMET predictor was obviously used to derive ocular permeability. Are there any specific in vitro tests that can mimic permeability in ocular tissues, or is this entirely in silico derived properties? Then there's a follow-on. So I'll let that one go first.
Yeah, that's a great question. Maybe if I take a stab at it, I think the best physical chemical parameter that I have actually personally measured is the partition coefficient. That is extremely predictive of corneal permeability, log P or log D. And then also, if you want to do some in vitro cell-based assays, I think there is a great correlation between CACO2 cells and corneal permeability, comparing two or three different models. But I think CACO2 is just so much more common. Almost every compound gets screened through that, so it's possible to translate that to some sort of a corneal permeability predictor. If you have any other ideas, chime in, please.
No,
Max, do you want to?
I think it's great already. I'm not aware of any specific testing. This is a question we see a lot, and I actually hope that the groups in academic like yours, Hovik, will come up with more solutions on the in vitro side to help us parameterize this model, because the predictions we obtain on the permeability side tends to work relatively well, but it's quite common that we have to modify a few when we are validating the model. So if we can come up with in vitro assays to help us parameterize the model, it will be very helpful to do a priori predictions when we don't have any data.
A question for you, Max, and putting you on the spot. I don't know how much you care to share, but that's my job. Obviously, since the inception of the OCAT model, there's been a lot of progress made, both in collaboration with and with the FDA and other agencies and other institutions. But where is the future? Where do you think that it should go next, and or where is the greatest need for data? I think you touched on it, but where do we need data to advance the model?
Yeah, so I think we're going to talk more about like all of the more complex formulations, insert, implants, the academic world and the biotech world, or limitless in terms of innovation that what they can create. So that's going to be one angle. The other angle is going to be probably the disease model. For example, how certain diseases affect the blood circulation in the retina. What could be the consequences on the PK? Because right now we focus more on the healthy side. And when we looked at the disease side for human for solution, we identified that there were no consequences. But there may be some diseases that have more significant impact on the physiology and that could impact the PK, but we could integrate that into the model to predict that as well. So the disease impact is going to be one angle to look at in the future.
Hovik, do you have a perception?
I agree. I completely agree with what Max said. You know, I think just the tip of the iceberg he presented today on correlating intraocular pressure modulation with ocular exposure. I mean, something like this has never been done before in my career, even. Nobody thinks that there is a way to do PBPK modeling, or pardon me, PK-PD modeling in in ophthalmology. PK-PD. This is going to be huge. Yeah.
That's great. Go ahead, Max.
No, I hope so that we'll be able to do it.
All right. I don't have any further questions in the queue. However, I did want to extend an invitation to anyone on the call that may not yet have access to the OCAT module or the module within GastroPlus, or entirely not have access to GastroPlus that might be seeking a trial license. We'd be more than happy to provide that so that you can get your hands on either the module and or the entire platform. Just give us a call or let us know, and we'd be happy to do it. Max, I'll throw it to you and Hovik for final comments.
Well, that's it. I think if you have just any question on the OCAT and how it can be used for preclinical and clinical predictions, like reach out to us. And if you want to partner with some new in vitro assays, reach out to Hovik.
Absolutely. I'm here. My door is open. Don't be shy. I think especially if you want to study these concepts, I love to share my experience and train interested students in drug discovery, in modeling informed drug discovery and drug development. I think this is the way to go.
All right, back to you, Jasmine. Thank you very much, all. Go ahead, Jasmine.
Thank you all. I just wanted to let you know that we do have upcoming webinars on our website. You can find them at simulationsplus.com. I also put a couple links in the chat if you want to follow us on social. And then once again, this webinar has been recorded for playback and will be available on our website and YouTube channel. This concludes our webinar for today. Thank you, everyone.